right, so this is a continuation of Lab 1. We're going to talk about blood typing and the process of how to do this. So to start with, I just want to make sure that we, just do a little refresher. Understand that there's in the ABO system, which is all about antigen A and B, there's 4 blood types. We've got type A, type B, type AB, and type O. And these are all about the combination of only 2 antigens. I have an antigen A and an antigen B. They're the only ones. And so if I have antigen A and only A, that means I'm type A. If I have antigen B and only B, I'm type B. If I have both, I am AB. And if I have neither, then I'm type O. Now, there is another one that gets added on here, and it is the Rh factor. Now, in blood typing and talking about the Rh factor, it is just referred to as antigen D. I usually just put Rh, but it's antigen D. So again, in the whole scheme of things, there's only 3 antigens, A, B, and D, which again, D represents Rh. All right, so we can have 8 different blood types. I have type A, and it can be negative or positive. And it's simple. If the D, the Rh factor is there, it is positive. If it's not, it's negative. So this one, since I don't see D antigen on there, this would be A negative. All right, B, this would be B negative again because I didn't put one on there. But let's say I put one on this one, and I'll just make it look like that or whatever. This would be AB positive. Again, just giving you examples. So again, the big thing is just understand there's only 3 possible antigens, even if I include the Rh factor in everything. Only 3. So all blood types are a combination of these 3. So with that being said, we'll look at each individual blood type and talk about how we're going to handle it. I shouldn't say each individual blood type. I'm going to look at the blood types and how this would work. So remember, if I've got antigen A on my cell, and that means that I cannot come in contact with anything that has anti-A antibodies. All right? So that's, if they come in contact, we have a reaction. So what I would do, this little drawing here is kind of an attempt to show you what would be called a blood typing plate. All this is a little plastic plate that would have 3 little indentations. And usually they'll have the little mark here to tell you what each one represents. And we'll talk about what that is more often in just a minute. But even if it doesn't, this is the acceptable normal pattern. A's top, B's bottom, the Rh or D is off to the side. So if I want to find out what blood type this is, what I would do is take this blood and put it into each of these little spots. All right, so isolate this blood cell. Again, we're moving all the antibodies or whatever. It doesn't matter, but we're moving them. And I'm going to take them and I'm going to put them in each one of these little indentations, right? So I've got the same blood in each one of these. Now, the way I want to test it is I want to look to see if this has A antigens. I'm isolating it. I want to look and see if this has B antigens. I'm isolating it. And this is looking for the presence of the Rh factor or the D antigens. Now remember, if a antigen comes in contact with the antibody that doesn't like it, we have a thing called agglutination, a clumping. Right? This is supposed to look like normal cells. This is clumped cells. So we're going back to this. If I have the same type of blood in each one of these little spots, now I need to test to see what antigens are present. So I'm looking for clumping. So if I want to test if A is present here, I am going to put anti-A antibodies in this little spot. If I want to test for B, I'm going to put anti-B in here. And if I want to test for the Rh factor, I put anti-D antibodies in there. And I look to see if they agglutinate. So remember, if I've got anti-A, so this is an A, this would be officially an A negative blood type, because the only antigen I have on my cell surface is A. So if this was to come in contact with an anti-A antibody, this anti-A antibody would attach to this and cause agglutination. All right, so we're going back to this example. And remember, just to remind you, this is A negative on this. So that means the only antigen on this is this antigen A. And that, again, each one of these little indentations has the same blood type in it. So I've isolated it. Remember, there's only 3 antigens, A, B, and I'm going to put this as D, which we'll say is the Rh factor. So there's only 3. So all I have to do is take this blood and isolate it and test for each one of these individually. And it's either going to agglutinate or it's not. So when I put the antibody into this, I'm going to put each individual antibody into its spot. So anti-A I put in here. It's either going to agglutinate or it's not. If it does, that means A is present. If it doesn't, it means it's not. I'm going to put antibody or anti-B antibody in here. If it finds the B antigen, it's going to agglutinate. If it doesn't, it's not. I'm going to take the anti-D antibody. I'm going to put it in here. Its whole job is to look for that Rh factor, the D antigen, and if it finds it, it will agglutinate. If it doesn't, it does not. So in this, what would happen is, because this only has the A antigen, the only reaction, the only agglutination would be in this little indentation part, this little section, because this has A antigen, so the anti-A antibody will find it and it will clump up, it will agglutinate. The other ones are supposed to represent that it didn't. So if you see the reaction, that means the antibody found the antigen, so the antigen is present. If it doesn't react, then the antigen is not present. So on this, I tried to draw it to where it looked like it. I don't know that I like the way that looks. It's hard to tell the difference on that. So what I would do is do this. I just draw little X's. These X's means agglutination, right? So on this, if I'm looking at it, this tells me that the anti-A antibody found the A antigen, so it is present. So I know I have an A. I'll write it over here. Since I put this in here and there's no reaction, I know I do not have a B. Since I know I put the anti-D antibody in here and there's no reaction, I know I don't have the Rh factor. So the only antigen that I have is A, and so that means this is an A negative. Let's look at this one. So I've taken this one, I've put anti-A antibodies in here. There is a reaction, so I know I have an A. I'll write it down here. I've put the anti-B antibody in there. There is no reaction, so I don't have a B. And I've taken the Rh, the anti-D antibody, and put it in here. And it did find the Rh factor, so it's present. So that means this is A positive. I'm hoping you're following this. We make it so much more complicated. Again, there's only 3, and it's either is it there or is it not. If it is there, I can, that means it's on the surface. If it does react, if it's not, it's not. Let's look at this one. So anti-A is here. It did react. Anti-B antibodies found theirs. It did react. The anti-D did not react. So if I'm looking at it and I'm looking at the three possible antigens that are on the cell, I know I have an A, I know I have a B, and I do not have the D. So that means I have a blood type that is A, B, negative. Again, don't make it more complicated than it is. If it reacts, the antigen is present. If it doesn't react, the antigen is not present. So let's look at this one. All right, this just looks like a blank one. There's no reaction here. Anti-A antibodies did not find an A antigen. No reaction here. Anti-B antibodies did not find a B antigen. And the anti-D antibodies did not find the Rh factor or the D antigen. So I'm going to give you a second to think about what this is. So it doesn't have any of these on here. So this would be O negative. This blood has none of the possible three antigens. Here's another one. No reaction here, no reaction here. We do have agglutination in the D or Rh portion. So this is now O positive. Remember, we get all these things kind of complicated. There's only three possible antigens and only three possible antibodies. Remember to call the antibodies anti-whatever, and it just makes sense how they mix and match. It's like having three colored expo markers, three different colors, and there's a cap for each color. If the cap binds to, if it finds the color that it's designed to fit on, we have agglutination. So I can have a black, a blue, and a red, and that's it. They're only one. So if I put the blue into a mix of it and it finds the blue pin and it caps onto it, that means that there's blue there. So if I take it like this, let's look at a few things and go back over. First of all, let's go back to a blood type. If I drew this here, and again, forgive the bad drawing because of the fact that trying to do this on a laptop really is not good. So I have agglutination in this one, and I have agglutination in this one. So that means that there is an A. So does this one have an A antibody, or antigen, I should say? It does not, so it's not this one. Does this one have an A antigen? It does, so it could be this one. This one also has an A, so it could be this one. This one does not have an A antigen, so it can't be this one. So now I'm looking at the B. It does not have a B. Since this one does have a B, I know it's not this one, so I'm left with type A. And so now I just look at this Rh factor over here, and since it did agglutinate, I know the overall blood type is A positive. Now the last thing I'm going to look at, I'm just going to go over the recipients and donors one more time. So in this, if I am looking for a donor, so I'd always try to draw my D's this way. So if I'm type A and I want to find out who I can give my blood to, who I can donate to. So if I'm type A, this is the blood that I'm trying to put into other people's bodies, and it has an antigen A on it. So I cannot put this blood into anybody's body that has the anti-A antibody. So in donating, you think about your blood, your antigen, and the other person's antibody, and they can't mix. So I can give blood to A because the A does not have anti-A antibodies. B does, so I can't give it to B. AB does not have any antibodies, so I can give it to AB. And O does have anti-A antibodies, so I can't give it to them. So if I am type A, the only people I can donate this blood to is A and AB. Man, I'm trying. If I'm type B, I'm going to go through the steps. If I'm type B, who can I donate blood to? I look around and I think, all right, since I'm type B, I know I have B antigens on my cell. I cannot give them to anybody that has anti-B antibodies. A has anti-B antibodies, so it's a no-go. I know B doesn't. I can give it to B. I look at type AB, again, no antibodies, and so I know I can give it to AB. And O has an anti-B antibody, so I can't give it to O. So if I am type B, I can donate my blood to B and AB. If I'm type AB, again, I look at my antigens. I've got antigen A and antigen B, so I cannot give it to anyone with either an anti-A antibody or an anti-B antibody. Since the other three, all of them have one or both of the antibodies, the only person AB can give to is other ABs. So if I go now to O, I look at my antigens that are on my cell, and since O has no antigens on the cell, I have no antibodies I am worried about, so O can give to anybody, and that's why it is called the universal donor. O can give to anybody. Now, if we want to be really specific, it is O negative that is the universal donor, because O negative can go into anybody's body. If I was O positive and I went into any of the negative blood streams, I would have that reaction to create the anti-RH or anti-D antibodies. And then the second time that happened, there would be a problem. So it is O negative is the true universal donor. So now if I'm looking at recipients, once again, now I'm going to change my look on things. If I'm a recipient, I'm not worried about the antigens on my cell, except for the fact that knowing them tells me what antibody I'm going to have in my system. So if I am type A, I know I have anti-B antibodies in my system. Because I start off with A, I am lacking the B antigen. So B antigens are my bad guy. They are the ones that I do not like. Right? The way I would look at this is to go through step by step. If I am type A, I have anti-B antibodies. I cannot receive blood from anybody with a B antigen. So anybody that has a B in the name, I can't receive blood from. So I can receive blood from A's and I can receive blood from O's. If I step it over to B, I've got a B antigen, which means I do not have an A antigen. So that means I have anti-A antibodies. I cannot receive blood from anyone with an A in their name. So I can receive blood from B's and O. Get it? B-O? Sorry. I know I ought to quit that, but just can't help it. It's this online stuff kind of gets me anyway, so I want to try and keep things somewhat jovial because it makes me want to scream sometimes. So anyway, now I get to AB. So if I'm type AB, again, I'm not worried about my antigens except to understand what antibodies I have, because antibodies are the key. If I'm receiving blood from someone, my antibodies will tend to attack it. So that's what I'm looking for. And since type AB does not have any of the antibodies, AB is considered the universal recipient, because it can receive blood from anybody because there are no triggers on it. Now, here is the thing. The true universal recipient is not just AB, it is AB positive. Because if this also has the RH, the D antigen, the RH antigen, that means there's nothing in my body that would surprise it, because it has all three of the possible antigens. So there's no antigen that would come in that would release an attack. So now if I step it over and look at O, if I'm type O and I need to receive blood from somebody, again, I'm not worried about my antigens except to understand what antibodies I have. And so I'm type O, so I have both antibodies. And since I have both antibodies, the only person I can receive blood from are other O's. And so that's the way it works. I hope you're following this. I hope it makes sense. It is a weird thing of how easy it actually is, but how easy it is to confuse it and get tripped up. Make sure you understand the foundation of it. There's only three antigens, there's only three antibodies. Whatever the anti-blank antibody is named, it doesn't like the other antigen, so it's going to attack it. So anti-A antibodies will always attack A antigens, and so forth. The other thing, just kind of to point out, everything on your body functions in a use it or lose it basis. And so red bone marrow is constantly trying to produce red blood cells. And as you age, your red bone marrow starts turning more to yellow bone marrow, which is a problem. If you get in a habit of occasionally donating blood, it makes your body remember that it needs the red bone marrow. The more it's used, the more it keeps it. The other thing is, it is always good to donate blood because it helps everybody. If you're type O, you can only receive blood from O. So if I'm type O, I want to give blood as much as I can because I can only receive blood from O. Now, they're going to use it as an alternate for other people, but that's a good thing. I want to continually put it in. If I'm type AB, I am the universal recipient, right? So I can only I can receive blood from anybody, but I can't donate blood to anybody else. But that helps me keep my red bone marrow healthy. So just a little FYI. Anyway, I hope that makes sense. I left this page blank just in case I needed to draw a few things on here. I hope everybody is having a great day, and I look forward to continuing all of this, and we will go from there.